All right, so thanks everyone for joining our session. I am very excited about the speakers that are going to be showing us their work today. So overall, it's been an amazing time for the care of cystic fibrosis patients. So these new therapies are really making strides in keeping our patients healthy. But I will say we are not done. Um, uh, as you may know, not everyone is eligible for some of these therapies, and they require to be given for a long time. So new approaches are needed to really reach the cure of this disease. And one of the most exciting candidates is the use of oligonucleotide or nucleotide-based therapies. So I think it's really important to understand all of the development in this field and the different approaches that different groups are taking to try to deliver these cells, these therapies into the right cells. Okay, so today we are very lucky to have Dr. Sylvia Creda, Dr. Kirill Pinol, Dr. Michael Torres, Dr. Alexandra Piotrowski, and Dr. Catherine X. Coffin, okay, who are going to be showing us their work. And we are going to start with Dr. Sylvia Creda. Okay, so Dr. Sylvia Creda is really a star in the field of cystic fibrosis research. She is an associate professor of medicine, biochemistry, and biophysics, and a member of the cystic fibrosis research group at the University of North Carolina in Chapel Hill. And Dr. Creda will be talking to us about correction of CF splicing mutations with oligonucleotides. Thank you very much for joining us today, Dr. Creda.
Good morning. I'm Sylvia Creda, an Associate Professor of Medicine in the Cystic Fibrosis Center at the University of North Carolina, Chapel Hill. I would like to thank the workshop organizers for giving me the opportunity to present our novel therapeutic oligonucleotide strategy to correct severe CF splicing mutations. These are my disclosures and grant funding. CF is a multi-organ disease with severe lung manifestations. Multiple types of Cftr mutations cause CF, and today approximately 90% of them can be overcome with a novel type of drugs named Cftr modulators. However, some CF patients with severe splicing mutations do not respond to current therapeutic modulators. Based on published in vitro data, it is possible to design oligonucleotide-based therapies for these individuals. Oligonucleotides are very short, heavily modified nucleic acids. The beauty of therapeutic oligonucleotides is that, unlike small molecules, oligos can be chemically similar but be rationally designed, sequence-specific for one particular mutation. They can be administered naked as drug-like compounds. They have relatively low toxicity and minimal genomic effects. And oligos are very stable and can have prolonged in vivo activity, reducing administration dose and frequency. Oligonucleotides offer a faster, safer therapeutic alternative compared to gene therapy modalities. And the last 5 years have seen the first successful clinical uses of oligonucleotides to correct genetic disorders. How therapeutic oligonucleotides work? There are different oligonucleotide modalities. For example, there are splice switching oligos or SSOs, which correct splicing mutations by modulating RNA splicing. In this talk, I will introduce our exciting data using SSOs to correct CF splicing mutations. During normal CFTR splicing, the spliceosome recognizes canonical sequences in the beginning and ends of exons in the pre-mRNA. The introns get excised, while the exons get joined together into a continuous coding sequence, which then gets translated into a normal protein by the ribosomes. However, a CF-causing mutation, such as 3849 plus 10kb, can create a new splicing site. And thus, an intronic sequence can be included as a pseudoexon, creating an aberrant mRNA that could be degraded and fail to produce CFTR protein. With little or no CFTR protein, modern modulators cannot be effective. In contrast, sequence-specific splice switching oligos or SSOs can be designed to align to a complementary sequence in the pre-mRNA and modulate the spliceosome into process correctly the mutant pre-RNA into normal RNA, which then can be translated into normal and active CFTR protein. The main obstacle to oligotherapies has been delivery of oligos to extrahepatic tissues, particularly the lung. Delivery is a three-part problem. 1, extrahepatic biodistribution and plasma membrane translocation. 2, endosomal escape, since more than 90% of the intracellular oligo is trapped and disabled in endosomes. 3, achieve efficient delivery with negligible toxicity. Our therapeutic approach attempts to overcome these barriers, particularly in the lung. Our strategy combines an SSO, which is chemically a peptide morpholino or PPMO, for improved organ and intracellular distribution, plus a small molecule with endosomal permeability capabilities named OAC. This combination allows to reduce the effective dose and thus the toxicity of each component. We designed a SSO specific for the CF3849 plus 10 kb splicing mutation with peptide morpholino chemistry to correct this mutation and rescue normal CFTR activity. We use our novel combination of SSO morpholino plus OAC and successfully corrected the splicing mutation 3849 plus 10 kb in bronchial epithelial cells derived from a patient homozygote for this mutation. These cells do not respond to novel modulator Trikafta, as shown in these graphs depicting CFTR channel activity. In contrast, our novel combination of PPMO plus a small molecule OAC significantly corrected CFTR activity, more than 30 microamps per square centimeter, the equivalent to normal CFTR activity in these cells. Our oligo plus OAC combination was effective when delivered as one-time treatment via the basolateral side and also the apical side of the epithelium with negligible cytotoxicity. Next, we tested our oligo plus OAC strategy in vivo. We use a reporter mouse expressing inactive EGFP interrupted by an intron harboring a splicing mutation. The mutant mouse does not produce EGFP, but when treated with a sequence-specific SSO, EGFP is expressed in any tissue the oligo is properly delivered. These are the highlights of recently published in vivo data in the lung. Both the SSO and the OAC were administered intravenously. The left panel shows effective splicing correction as high levels of EGFP expression in the bronchial epithelium and the muscle layer of mice receiving both the oligo plus OAC. Epithelial correction was observed in both ciliated and non-ciliated cells, such as secretory and basal cells. The graph on the right illustrates mRNA analysis after one in vivo treatment with oligo at 5 milligrams per kilogram of body weight plus OAC. Efficient correction was achieved with approximately 80% correction to wild-type mRNA, and expression of wild-type mRNA persisted for 22 days in the lung after one treatment. The treatment was well tolerated with no indication of toxicity. Efficient correction was also achieved in other CF-relevant tissues, such as intestine and liver. One systemic treatment of SSO plus OAC significantly corrected HEFP, which was clearly observed in freshly excised intestine and fixed liver sections, shown in the left panel. On the right, the graph quantifies the percentage of correction of mutant mRNA into wild-type mRNA with one treatment of Oligoplus OAC after 2 days post-treatment, and correction persisted for 22 days, the length of the experiment. We also tested pulmonary delivery of oligonucleotides via intratracheal instillation of oligo alone. The left panel describes bronchial HEFP expression. In the right, the graph depicts the significant level of mRNA correction with one treatment of oligo at the concentrations of 1, 2.5, and 5 milligrams per kilogram of body weight, with no evidence of toxicity. In a related project, our lab has a partnership to develop a novel aerosol system to deliver different modalities of oligonucleotides. The system, designed originally by Hickey and collaborators, used a hot wire embedded with lipid and a therapeutic molecule. In our studies, we use a therapeutic SSO. During heating of the wire, the lipid propels and envelopes the oligo into particles which are formed in situ by condensation. The particles have a range of sizes, including those productive for lung therapeutics. Using live cell cultures expressing the reported HEFP system, we tested the hot wire technology. Particles of size smaller than 1 micron and 1 to 2 microns produce oligo deposition and significant HEFP correction and expression. Our initial in vivo experiments in mice aerosolizing a blue probe with this novel system shows promising deposition in the airways. In conclusion, our data shows that combining a therapeutic oligo with an endosomal compound allows for relatively lower doses and prolonged effects correcting a CF splicing mutation, minimizing toxicity. Our oligonucleotide-based strategy offers a novel therapeutic platform for CF patients who do not respond to current modulators. Some of the data presented here have been recently published in Nucleic Acid Research. The poster 598 is associated with this presentation. Thank you very much. I'm happy to answer any questions.
All right, excellent. Thank you so much for that presentation, Dr. Creda. So we have a few questions in the system. So the first question is, how does SSO activity last so long in the cell? How long does it last in vitro in HBECs? And please unmute yourself.
Hello, thank you for the question. These oligonucleotides are heavily modified and they are very stable. In vitro, we have some of our studies up to 6 months, essentially the life of the cultures. And we are running now a longitudinal study with some cells that we start to treat in February of this year, and they still have HEFP expression. So this is with our reporter system. Mm-hmm. In vivo, we believe that we can reach 2 months. We are running currently this study, so we know for sure we have very good expression for 1 month after only 1 treatment. Okay, and the other thing that we are doing is repetitive treatments. In the clinic currently, some of these oligonucleotides in other applications, not in CF, they are dosed between 3 and 4 times a year. And this is, thank again, they are very, very stable once that they are in the cell.
Very good, thank you. So the next question is there any off-target activity for other genes?
This is a very important question. Most people do it. Actually, it's very rare. Although they are short, particularly the oligonucleotides that are target to intronic sequence. And usually these intronic sequences are not canonical. So they are very specific, and it's a very, very low probability to have an off-target effect or any other genomic activities or activities against the mRNA. However, it's always very important, and is part of the preclinical studies for every oligonucleotide therapeutic approach, to study putative off-target effects, both at the genomic and the transcriptome levels.
Great, thank you. The next question is, as I understand it, the in vivo work used GFP. Have you tried to correct a membrane protein?
This is a very good question, not at this time. So we essentially use this reporter mouse for all our dose and toxicity preclinical studies. And we did obtain a CF mouse from Dr. Dram, but it's very difficult to use it in vivo. We are trying to have some in vitro models derived from this mouse.
Thank you. The next question is, does OEC compound promote release of molecules or particles larger than SSOs from the endosomes?
Yes, and it's actually published. So they are essentially increase temporary the permeability of the endosome. So many molecules in the endosome can escape. So it can be applied to other nucleic acid therapeutic approaches.
And then next question is normal endosomal recycling of membrane proteins affected by the OEC?
We didn't address that particularly. We did general toxicity studies, and we used them as subtoxic dose for a very short period of time, because anything that essentially will increase permeability of the endosome, carry the problem of, a potential problem with toxicity. So the quick answer is we didn't address it directly, but we did general toxicity studies, and doesn't affect greatly the biology functions of many proteins.
Thank you. And a related question for the toxicity that you evaluated, what type of toxicity was observed?
For the endosomal compounds or the oligonucleotides? In
general, for the oligonucleotide and the compound. Yep.
So the oligonucleotides had a very good safety profile. And we acquire activity, much lower than, subtoxic concentrations. For the endosomal compounds, we have two prototypes from two different chemical families, and they do have a narrow therapeutic profile. So again, they need to be dosed at very low doses for a very short period of time. The advantage to do two components, oligonucleotide plus endosomal compound, as I indicate in the beginning, is that you can essentially fine-tune both components and use them at very, very low doses and have a very good therapeutic activity.
Good, thank you. And our last question. What is known about the mechanisms of differential efficacy of the SSOs apically versus basolaterally delivered in the ALI cultures? Like, for example, Could different efficacy be due to surface area or differential rates of endocytosis or some other mechanism?
Very good question. Actually, when we submit the paper, we have a similar question from the reviewers. I would say all of the above. It's very difficult, essentially, to estimate an equivalent dose in the apical and the basolateral side. So you have two very different surface areas in the apical versus the basolateral. And also in the case of airway liquid interface, it's very difficult also to control the volumes and therefore the total mass that you apply to either side. So you can, have an estimation of what, compare the dose in the apical and versus the basolateral, but it's very difficult. And it is possible that in our studies, the difference between the apical and the basolateral is due to a major dose in the basolateral side for all, not just only the surface, but also the basolateral side had a very, very active endocytosis activity.
Okay.
Hello, everyone. My name is Kiriel Pino, and I am a postdoc in the NHLI at Imperial College. I would like to thank the organizers for inviting me to share my work on our efforts to develop molecular assays to determine gene transfer efficiency for an upcoming first-in-man clinical trial using a lentiviral gene therapy vector for cystic fibrosis. And these are the relationships that exist for this presentation. We have a financial relationship with Boehringer Ingelheim, and our lentiviral vector is manufactured by Oxford Biomedica. And so with our partnership with Boehringer Ingelheim and Oxford Biomedica, the UK Gene Therapy, Respiratory Gene Therapy Consortium is working to develop a pseudotyped lentiviral vector for pulmonary gene therapy applications like cystic fibrosis. And we are currently progressing towards a first-in-man clinical trial where our gene therapy vector carrying a CFTR cDNA will be administered via an aerosol to the lung epithelium, where it will be able to integrate and restore CFTR expression and function in the lung epithelium. So in this clinical trial, in addition to the clinical disease outcome measurements, one of the key outcomes for success is trying to quantify expression of the delivered gene therapy product. So this would be molecular detection of expression of restored CFTR in the airway epithelium. We anticipate that we will be able to collect epithelial brushing samples from our trial participants for these analyses. Now, in the past clinical trials for cystic fibrosis gene therapy, we and others have used PCR and RT-PCR from DNA and RNA extracted from these bulk airway epithelial brushing samples. However, a major drawback from these types of assays is that these methods, while they give you information on if your product is expressed at all, they cannot actually provide information on how efficiently your gene therapy vector has transduced the airway epithelium on a cellular level. And so we have been working to refine our bulk PCR and RT-PCR assays to try and develop assays in small cell pools and at the single cell level to quantify our vector transduction efficiency. And for these experiments, we've been using human air-liquid interface cultures. So these cultures are from human bronchial epithelial cells, and at the undifferentiated stage, we've transduced these cells with our lentiviral vector, either carrying a EGFP reporter or the cystic fibrosis CFTR transgene. After transfection, we have airlifted these cultures and allowed them to fully differentiate. Now, from cultures that were either transduced with our GFP or CFTR carrying vector, we have generated bulk, small cell pools, and single cell samples for assay development. And the added bonus of using our eGFP reporter is that we're also able to quantify transduction efficiency using flow cytometry, which provides a benchmark for evaluating how successful our molecular assays are. So to sort of benchmark our study, we began by doing bulk molecular characterization of our ALIs transduced with either our GFP or CFTR vector. On the left, you can see a PCR assay looking at integrated vector copy number. Now, I should pause here and mention that for all of these studies, we are using a primer combination that recognizes sequence that is present in all of our lentiviral vector constructs. So it's the same primers that are used to characterize both cells transduced with either our GFP construct or the CFTR construct. So again, on the left-hand panel, we are looking at integrated vector copy numbers. So this is showing lentiviral vector that has been reverse transcribed into cDNA and integrated into the genome. And we can see that there is no difference in the frequency of vector integration between our GFP or our CFTR transgene constructs. What we were surprised to see is that there is approximately 1 log lower transgene mRNA expression, shown here on the right, of the CFTR transgene compared to the eGFP. Now, there's a significant difference in the transgene length of CFTR compared to eGFP, and we still don't know if this decrease in transgene expression is due to the difference in size and actually expression of the two constructs, or if it could be perhaps due to mRNA stability of the larger transgene. However, when we looked at expression of the transgene-derived CFTR in the middle panel, we're calling it the codon-optimized CFTR, compared to the endogenous human CFTR shown on the left, we can see that we get 10 times greater expression of the transgene compared to the endogenous expression, which is very reassuring when we think about the clinical utility of this transgene construct in CF patients. Next, we wanted to try and quantify the transduction efficiency, and we first stopped from bulk samples down to 50-cell pools. And to begin to develop this assay, we first started with a A549 cell line, either the wild type cell line or a cell line that has been transduced with GFP. And we created 50-cell pools using FACS and controlled numbers of GFP positive cells and GFP negative cells per pool, and performed RT-ddPCR to look for mRNA expression of GFP. What we can see is that these 50-cell pools can successfully detect rare positive events within the sample, down to even 1 or 2 cells out of 50. And when we mathematically try and estimate the number of GFP positive cells per pool, we see that there is great correspondence between the known number of cells per pool and the estimated number of cells based on GFP expression, which reassures us that this assay is going to be very useful in our experimental samples. We then moved on to our ALI samples that were transduced with our GFP or CFTR constructs and analyzed our 50-cell pools. And these small cell pool samples basically reflect the bulk RT-ddPCR data, showing a reduced expression of CFTR compared to the eGFP mRNA. However, what you can notice in our CFTR transduced samples is that there's a small number of pools where we see elevated copies, and this reveals the utility of using this small cell pool assay compared to just the bulk assay in being able to detect infrequent positive events. And these assays actually get us one step closer to being able to begin estimating transduction efficiency, as we can assess that any pool that is above the threshold of the assay contains at least 1 positive cell. And from there, we can know how many pools of 50 cells that we have and the conservative estimate of at least how many pools contain 1 or more positive cells. And finally, we refined our assay one step further and developed it into a single-cell RT-ddPCR assay. So for these assays, we again used our ALIs transduced with either our GFP or CFTR constructs, and we first quantified the transduction efficiency of the GFP transduced samples by flow cytometry. From there, we used FACS to generate sorted single cells, and we generated known GFP positive and GFP negative cells as controls to measure the sensitivity of the assay. And then we also generated plates of single cells that we're calling unbiased live cells. And what we mean by that is that we did not sort them based on any sort of fluorescence activity. It was just, if it's a live cell, it landed into one of our pool or our samples that we could analyze. And when we looked at our assay sensitivity, we showed that at least 70 or greater than 70% of cells that were GFP positive by FACS could be detected by our RT-ddPCR assay. And additionally, we noticed, now that we looked at single cells, that there's a wide range in expression of the transgene. So a 10-fold range in transgene copies per positive cell. And this type of information, again, you could not get from the bulk DNA and RNA assays. When we then went and quantified and looked at our GFP versus CFTR transduced cultures, we first, looking at our GFP samples, saw good consistency in the percent transduction efficiency measured by flow cytometry and estimated by our RT-ddPCR single cell assay. So both assays estimated about 13% of the cells within the sample were transduced. When we then compare the GFP and CFTR cultures at the single cell level, we can see that there's a similar range in transgene copies per cell, which is reassuring that the CFTR vector is behaving similarly to our reporter gene. However, similar to the outcomes that we saw in the bulk and 50-cell pool assays, we estimate a reduced transduction efficiency of the CFTR vector compared to the GFP. However, if you think back to the earlier data that I showed you, showing that the integrated vector copy numbers between the two vectors were the same, it's entirely possible that the CFTR is not transducing at a reduced efficiency, but perhaps that there are positive events that are falling below the assay threshold from these single cell assays. And now if we move back to the clinical utility of these types of molecular assays, and we put them into context, your bulk DNA and RNA assays are going to be a great starting point to assess the global transgene expression of your therapeutic target. And they're also a very high throughput assay. They're easy to perform, and you can perform it rapidly on lots of samples. We then move and refine a little bit further to our small cell pool assay. This type of assay will allow you to detect rarer positive events within your sample, and you can begin to estimate the cellular transduction efficiency of your vector. However, this type of assay requires a bit more upstream processing and is more of a medium throughput assay. And if we refine even further down to the single cells, this type of assay, if you analyze sufficient numbers of single cells, can give you an exact cellular transduction efficiency measurement. And it gives you the added information that you know what the expression of your transgene is at the cellular level, and it lets you understand how similar the transduced cells are to each other. Are they expressing similar copies, or do you have a very wide range of expression on a cell-to-cell basis? However, this type of assay requires a significant amount of upstream processing and requires processing of a lot of cells to get you an accurate cellular transduction. So it's a much lower throughput assay. And as we're thinking about moving into our first-in-man clinical trial for our lentiviral vector in cystic fibrosis, we anticipate continued development of all three types of assays as we move towards assessing the success of our gene therapy product. And with that, I would like to acknowledge the entire gene therapy group in the NHLI at Imperial, particularly my supervisors, Eric Bolton and Uta Griesenbach. I would like to thank as well our collaborators at Boehringer, Sebastian Krutz and Elena Moisenko, as well as our funding sources. Thank you very much.
So that was actually Kirill Pinol, who is a research associate at Imperial College London. So thank you for showing that data, Dr. Pinol. There's a good number of questions in the system. So let's start with the first one. Katex Coffin is asking, how does this level of mRNA correspond to functional correction in CF epithelia? Is codon optimized CFTR 10 times more than wild type?
Thanks. That's a great question. So what we know is that at this level of transduction, if we do a functional assay like an Ussing's chamber analysis, we can get somewhere, I believe, around 40% of wild type levels with this level of transduction. And then in terms of the codon optimized version, I don't believe we have evidence that it is supra-functional compared to the wild type. So that should be not changing the function of the channel, but simply the coding sequence of the protein.
Very good. Thank you. The next question is, what range of MOIs do you use for the ALI culture studies?
That's also a good question. So for all of the data that I showed you, it should be MOI 10, but we've gone down between MOI 1 to MOI 90, and you simply impact the transduction efficiency, and then we'll pick up more or less copies in our molecular assays.
Good, thank you. So the next question is, were the small cell pool and the single cell assays done with native or codon optimized CFTR?
Okay, so that's a good point. So the cells themselves were transduced with either lentivirus lentivirus carrying a GFP reporter or our therapeutic codon optimized CFTR. But the primer probe set that we use to actually detect the copies is for a element within our reporter cassette that's universal. So it was the same primer probe, so we weren't detecting codon optimized CFTR. Hopefully that answers your question.
Yep, thank you. Another question is, how long were your single cells cultured after removal from ALI before analysis?
Oh, that's also very good. So these cultures were transduced in the undifferentiated state, and then I believe they were airlifted for 3 weeks and then collected for analysis.
Got it. Thank you. Another question is, so in thinking about expression of your GFP and CFTR transgenes and the observation that the CFTR transgene had 10 times native CFTR expression, what is the promoter, polyA signal, and UTR sequences that you're using in your GFP and CFTR transgenes? Is your CFTR transgene codon optimized?
So the promoter and poly-A should be the same between the two constructs, and our promoter, we've published on this, so that can be checked if I get this wrong. It should be an HCEF promoter, but those full details are published. I apologize for perhaps a little lack of preparation on that granular detail. Thank you.
Yeah. Thanks for your answer. So next question is, have you looked or been able to determine the levels of expression in different cell types? For example, ciliated secretory cells via single cell RNA sequencing.
We have not done any single cell RNA-seq. What we know from our GFP construct is that all of the different cell types, all the major ones, let's say, so ciliated, goblet cells, etc., can be transduced by our vector, but we haven't done more granular than that. But that's an interesting question.
Got it. Okay. Next question. How long does the expression of CFTR last?
That is a great question. So we're using a lentiviral vector, which should be integrating, which means that if a cell's transduced, it should express it for the lifetime of the cell. And our animal studies show that we can get long-term, like lifelong in a rodent expression. So hopefully that gets at your question.
Okay. Thank you. Next question is, can you describe the relative success of transducing undifferentiated versus differentiated cells as will be challenged to do in vivo with your vector?
That's a great question. So we have published and shown in numerous animal models, as well as numerous animal models in vivo, that we can get good transduction of the airway epithelia. When it comes to the ALI cultures, I would say we perhaps have better success with our undifferentiated cultures, but the reasons why are unclear at the moment.
Okay. And then I think we still have time for a couple questions. Do you plan to use this type of assay on clinical samples?
Yeah, that's the goal. So at the moment, we're working to develop it to be robust enough and sensitive enough. And the question then becomes whether the single cell analysis on a patient is going to be financially and time-wise feasible, because it's very labor intensive, but the added benefit of knowing the exact transduction efficiency may make it worthwhile.
Got it. Okay. And then let's do our last question before we move on. Which media were you using to differentiate your cultures at ALI?
That work was done by our BI team, so I will not be able to answer that in full. Sorry.
Okay. All right. Well, thank you very much, Dr. Kirill Pinot. So now we're going to move on to our next presentation. So Dr. Sylvia Creda, as I mentioned a bit earlier, she's an associate professor of medicine, biochemistry, and biophysics, and a member of the CF Research Center at the University of North Carolina in Chapel Hill. Thank you for joining us today.
Hello, my name is Dr. Michael Torres, and I am the VP of R&D at RECODE Therapeutics. Before I begin, I'd like to thank the selection committee for inviting us to share our work toward developing an inhaled mRNA-based therapy for the treatment of cystic fibrosis. Here are my disclosures. I am a shareholder and employee of RECODE Therapeutics. RECODE is leveraging our platforms to develop two distinct approaches to address the significant unmet need in cystic fibrosis. Our lead program for CF is an mRNA-based therapy, which will be the focus of my talk today. In addition, we are leveraging our SORT LNP platform to enable full gene correction of basal cells that could provide a lasting therapy for patients. Our gene correction work will be presented at a later meeting. Here, we are providing more details of our mRNA approach. The initial focus will be on patients not currently amenable to CFTR modulator therapy. Our therapeutic cargo is CFTR mRNA. The nucleotide sequence is optimized to improve stability, quality, and translation efficiency. We use modified nucleotides for reduced immunoreactivity. To deliver CFTR mRNA, we use our proprietary lipid nanoparticle system optimized to carry messenger RNA and also optimized for transfection of pulmonary lung epithelial cells, and is compatible with nebulization. The formulation is delivered as an aerosol using a commercially proven mesh nebulizer. RECODE has designed and screened multiple CFTR mRNA sequences and modifications. We have compared the RECODE CFTR mRNA to the human wild-type sequence using the conductance assay to evaluate CFTR function in FRT cells. As shown in the bar graph and selected traces, we see significantly higher CFTR function compared to the wild-type CFTR mRNA. Our data indicates our MOD4 sequence is the most active CFTR mRNA to date. This is our current lead CFTR mRNA. We have evaluated our CFTR mRNA further in FRT cells. As shown by the traces, we see significant CFTR function that is dose-dependent and correlates with the amount of CFTR protein expressed. As mentioned previously, aerosol delivery is an essential feature of our mRNA-based approach. We use the Vitro cell exposure system coupled with a vibrating mesh nebulizer to evaluate nebulization feasibility and uptake of our formulations into primary CF-HBE cells that are fully differentiated and grown at the air-liquid interface. The images on the right demonstrate that we have formulations such as RTX0001 that can be aerosolized and efficiently deliver reporter mRNA, such as tomato red mRNA, into these mucus-producing primary CF-HBE cells. A unique feature of our SORT LNP platform is the ability to tune the composition to alter the cell tropism patterns. This slide shows examples of formulations we have optimized to deliver reporter mRNA into primary CF-HBE cells using the Vitro cell system that was described in the previous slide. Using the markers indicated on the table, we show formulations that deliver mRNA to therapeutically relevant cell types such as circulatory and basal cells. With SORT, one can optimize formulations to enhance delivery into the target cells of interest. In this experiment, we evaluated rescue of CFTR function after a single aerosol administration of RTX0001 in fully differentiated primary CF-HBE cells. CFTR function was measured 24 hours post-treatment using the TEC24 system to calculate the equivalent current. As indicated by the traces on the left, we see significant CFTR rescue after stimulation by forskolin VX770 that is comparable to a triple combination of CFTR modulators in these cells. In support of the function data, we observed significant levels of CFTR protein as determined by qualitative Western blot. This data demonstrates that our formulations can rescue in the gold standard model of CF. To support the continued development of our formulations, we have evaluated in vivo distribution and tolerability in mice. Using luciferase mRNA and bioluminescent imaging, we observed significant distribution and expression in the lungs of mice after aerosol administration. Additional studies have shown no significant impact on tolerability as measured by histopath cytokine upregulation and standard ClinChem readouts. In summary, we have shown that RECODE-designed CFTR mRNA sequences produce high levels of CFTR protein and function. In addition, we show formulations that can be nebulized and, importantly, transfect fully differentiated CF-HBE cells. Using our formulations to deliver CFTR mRNA, we see significant CFTR function in the gold standard model comparable to CFTR modulators. Ongoing activities include additional formulation optimization and dose finding in CF-HBE cells. We plan to test our lead formulations in genotypes not currently amenable to modulator therapy, such as W1282X homozygous cells. In addition, we plan to evaluate our lead formulations in single and repeat dose studies in mice as we continue the development of an effective inhaled mRNA therapy for the treatment of cystic fibrosis. Thank you for your time.
All right, that was great, Dr. Torres. Thank you for showing us your data. So we have a few questions in the system. So the first question is, does CFTR codon optimization in FRT cells carry over to primary HBE cells?
In our experience, the modifications and the rank order of mRNAs does in fact correlate with activity. Of course, there's an additional layer of delivery, which is a separate issue. But in the case of our most potent formulation coupled with our varying mRNAs, we do see a correlation between FRTs and HBE cells.
Got it. Thank you. The next question is nebulization with the Vitro cell required for entry and expression, or will addition as a liquid give the same amount or greater expression?
In our experience, we've assessed both. We call apical bolus or liquid bolus as our way to deliver as just a straight liquid. It tends to be about 50 microliters on the standard 24-well transwells that we're using. We in fact do see enhanced uptake with the aerosol, delivery by nebulization versus apical bolus. And so, again, carrying that forward to our target product profile, we tend to continue using aerosol delivery as our preferred route of assessing formulations and function in primary HBEs.
Good, thank you. Next question is, do you know which cells in the lungs of mice express the transgene?
So we have not looked at that in mice per se. We have it within our company. We have an additional program for primary ciliary dyskinesia, and within that system, we're developing multiplex IF panels that we'll disclose data for that at a separate meeting. But the comment there is with those MIF panels, we have done single dose studies in non-human primates and can see that we're hitting secretory cells, basal cells, so goblet cells as well by this aerosol method. So we do see, and there we're now actually going back to the mice to develop those panels, but we're seeing at least in non-human primates that we're able to hit the same types of cells in the same region, conducting airways in the lungs of non-human primates that would be of interest for us.
Got it. Thank you. Next question. Have you looked at the toxicity or inflammation induced by these preparations?
We have in a variety of different models in the HBE cells. We use various assays, LDH. We can look at cytokines. We do not see a significant increase in those readouts there. In mice, the histopath has looked clean, and no red flags from any of the pathologists, toxicologists that we've looked at. And again, in the NHPs where we are using masks, we've not had any significant red flags indicated in the same readouts, histopath, et cetera. So we feel we're in a pretty good shape with the current formulations that we're using, but obviously are continuing to optimize those.
Good. And then our last question, have you explored the mucus penetrating properties of this formulation? And is it going to be different in mice versus in humans?
So we are developing internal assays to look at mucus penetration. Again, in the primary HBE cells, we don't do, I would say we use standard washes to culture and maintain the cells, but no specific method to remove all the mucus before treatment or what have you. And then in future studies, we are going to be looking at the CF ferret model as well. Of course, the G551D ferret that John and the rest of the group have come up with in Iowa to further assess in a relevant setting with mucus biodistribution level. So we hope to have better answers in a variety of models, but to date, at least within the HBEs, we see that mucus has not been an unsurmountable barrier for us with using our formulations.
All right, thank you very much. So in the interest of time, we're going to move on to our next presentation. I wanted to remind all of the viewers that you can always contact the presenters at their poster or via their poster session system. Moving on, our next presenter is Dr. Alexandra Piotrowski-Daspit. who is a postdoctoral fellow in the Salzman Group at Yale University. Thank you for joining us today, Dr. Piotrowski-Daspit.
Hello. I'd like to start by thanking the organizers for this opportunity to share our work with you today. My name is Alexandra Piotrowski-Daspit, and I'm a postdoctoral fellow working with Mark Salzman, Peter Glaser, and Marie Egan at Yale University. My talk today is titled In Vivo Nanoparticle-Mediated Therapeutic Nucleic Acid Delivery for CF Treatment. The following relationships exist related to this presentation. I'm a founder of and consultant for Xanadu Bio. As we all know, Cftr encodes a chloride channel key to balancing ion and water secretion and absorption in epithelial tissues. In patients with CF, Cftr dysfunction causes mucus in several organs to thicken, leading to lung infections, a reduction in lung function, and also poor digestive function in the gastrointestinal or GI tract, highlighting the fact that CF has a significant Hello. I'd like to start by thanking the organizers for this opportunity to share our work with you today. My name is Alexandra Piotrowski-Daspit, and I'm a postdoctoral fellow working with Mark Salzman, Peter Glaser, and Marie Egan at Yale University. My talk today is titled In Vivo Nanoparticle-Mediated Therapeutic Nucleic Acid Delivery for CF Treatment. The following relationships exist related to this presentation. I'm a founder of and consultant for Xanadu Bio. As we all know, Cftr encodes a chloride channel key to balancing ion and water secretion and absorption in epithelial tissues. In patients with CF, Cftr dysfunction causes mucus in several organs to thicken, leading to lung infections, a reduction in lung function, and also poor digestive function in the gastrointestinal or GI tract, highlighting the fact that CF has a significant effect on multiple organ systems, and that an ideal therapeutic would ameliorate the disease in all affected tissues. Using a non-nuclease gene editing technology based on triplex-forming peptide nucleic acids, or PNAs, we have sought to correct the underlying genetic defect. PNAs are DNA analogs consisting of a charge-neutral peptide-like polyamide backbone with nucleobases enabling hybridization with DNA with high affinity. These can be designed to bind site-specifically to genomic DNA, and for gene editing purposes, can form PNA-DNA-PNA triplexes by invading genomic DNA and forming a bulky lesion adjacent to a target mutation site. PNA triplexes recruit the cell's endogenous DNA repair system, such as nucleotide excision repair and homology-dependent recombination pathways, to initiate site-specific modification of the genome when a single-stranded donor DNA is co-delivered as a template containing the desired sequence modification. We've also recently developed next-generation triplex-forming gamma PNAs containing a polyethylene glycol substitution at the gamma position, and these actually help promote the helical organization of the PNAs and promote enhanced DNA binding. In the context of CF, in prior work in a collaborative effort by the Salzman, Glaser, and Egan laboratories, PNA and donor DNA molecules were designed for the site-specific correction of the F508del CF mutation, encapsulated into polymeric nanoparticles primarily composed of polylactic coglycolic acid, and then tested in a murine F508 model in vivo. In this case, mice were treated with a nanoparticle suspension by an intranasal application, and after treatment, correction of the mouse Cftr mutation in the nasal epithelium was assayed by measuring the nasal potential difference in vivo as a non-invasive assay. After intranasal delivery, the impaired response to Cftr stimulation was partially corrected, with mice exhibiting NPDs that hyperpolarized, which is more characteristic of wild-type mice. And so moving forward, we have sought to expand the gene editing potential of our PNA-DNA nanoparticles by delivering them systemically to reach other organs apart from airway epithelia that are also affected by the disease. Moreover, we wanted to incorporate the modified gamma PNA chemistries, as these have shown enhanced binding in other disease models. And so based on previous work, we designed tail clamp PNA molecules that bind near the mutation site with homopurine stretches. Here, we incorporated modified gamma PNA monomers into the PNA sequence in the Hookestein domain, where PNAs are thought to bind initially. PNAs are hydrophobic and not readily taken up by cells, and so we thought to improve delivery by encapsulating these gene editing agents into polymeric vehicles using the biocompatible PLGA polymer, and use these vehicles for both in vivo and in vitro delivery. These nanoparticles formulated with PLGA are spherical in morphology and about 240 to 270 nanometers in diameter with a negative surface charge. And here on the right, I'm showing an SEM image of what these nanoparticles look like. STF is a systemic disease with a heavy burden on both airway and GI epithelia. We first tested whether PLGA nanoparticles could reach multiple organs when administered systemically. And so we performed an in-depth biodistribution assessment of these vehicles over the course of 48 hours using whole organ fluorescence by IBIS imaging, as well as flow cytometry of different cell types in several tissues of interest. And while the majority of nanoparticles do accumulate in the liver, we also observed nanoparticle accumulation in both the lungs and also the GI tract, which was exciting to us. And importantly, we see accumulation in multiple cell types in the lung, including epithelial cells. And so next, we wanted to assess the efficacy of our PNA nanoparticles in vivo. And here I'm showing our systemic delivery treatment scheme. After baseline phenotypic CFTR activity measurements, F508 mice received 4 doses of PNA nanoparticles spaced 2 days apart, after which we assessed post-treatment CFTR activity and also gene correction. 2 weeks after the last treatment, in vivo phenotypic correction of the F508 mutation was assessed using nasal potential difference, and in this case, we also looked at rectal potential difference. In our studies, after 1 dosing round of IV-delivered gamma PNA nanoparticles, we observed a significant amelioration of the impaired response to cyclic AMP stimulation in the nasal epithelia, and also a slight response in a subset of animals in the rectal epithelia. When we look at pretreatment and post-treatment PD responses for each mouse, we observed a decrease in NPD for the majority of gamma PNA nanoparticle-treated mice, whereas the majority of blank nanoparticle-treated mice exhibited no change or often an increase in NPD after treatment compared to baseline measurements. A similar analysis of RPD responses shows a decrease in RPG for the majority of gamma nanoparticle-treated mice and no change or an increase in RPD for blank nanoparticle-treated mice. We also assessed gene correction by ddPCR in several organs. We were able to detect editing in tissues that make up the airways and GI tract, with higher levels of editing observed in the airway epithelia, particularly in the nose and trachea, but also some editing detected in the ileum, colon, and rectum, and higher levels of editing correlated with improvements in phenotypic response. While we were excited about these results and our ability to deliver nucleic acids using simple polymeric systems such as PLGA, we're also striving to produce new materials that are even better at nucleic acid delivery. In the context of PNA DNA gene editing agents, for example, we would like to improve our in vivo gene editing efficiencies by enhancing nucleic acid loading and transfection efficiency, as well as other nanoparticle characteristics, which can in turn influence cellular uptake and also biodistribution. Now, in vivo delivery is actually quite a complex problem with barriers present at multiple levels, including the organism level, the tissue level, and even sort of intracellularly at the cell level. And a lot of traditional preclinical therapeutic screening is done in 2D cell culture, but it's been shown now that these kinds of studies are just not very predictive of what actually happens in the in vivo environment, highlighting the need for better models and standards with which to evaluate therapeutics. We're interested in understanding what structure-function relationships determine the physiological fate of nanoparticles. In particular, we would like to understand the effects of multiple parameters on in vivo delivery, including polymer and nanoparticle characteristics, dosing, and even the strategic use of co-injectable agents to enhance biodistribution to target tissues. One particular family of materials that we are interested in for this purpose is polyamine coesters, or PACEs. The basic or classic polymer in this family has 3 component monomers: a lactone that confers hydrophobicity and stability, an aminodiol that confers a mildly cationic charge, and an ester that enables additional hydrophobic control or variations in end group chemistry. And there are several reasons why these polymers in particular are promising for nucleic acid delivery. Like PLGA, they're biodegradable and biocompatible, and due to the incorporation of amine-containing monomers, they're also mildly cationic, which makes them less toxic than other heavily cationic polymers, but also enhances encapsulation of negatively charged nucleic acids. These materials are also highly versatile and can be formulated into different kinds of delivery vehicles, and it's easy to modify both their chemical composition and their physical properties. And so we can synthesize quite a large library of these materials and formulate them into vehicles that we've shown to be effective for the delivery of a wide range of nucleic acids, including plasmid DNA, mRNA, and siRNA, both in cell culture and in vivo. In a recent report, we developed a high-throughput evaluation tool to standardize and accelerate the analysis of circulation half-life of intravenously injected therapeutics. A schematic of this workflow is illustrated here. After intravenous injection of any fluorescently labeled agent, such as diluted polymeric nanoparticles, 2 microliters of blood are collected at desired time points following injection. After blood collection, blood samples are dispensed into a multi-well glass bottom plate along with a set of standards. And then images of each sample are then acquired using an automated imaging system. Fluorescence intensities of these images can then be fed into a custom MATLAB program and compared to the standard curve to obtain the concentration in blood for each sample. Importantly, because we're sampling a total of less than 1% total blood volume, other experimental endpoints can also be measured during and after blood collection, such as flow cytometry or whole organ imaging for biodistribution assessment. In the context of CF and lung delivery in particular, we're also interested in studying uptake in different cell types in the lung. And so we've also developed flow cytometry protocols to be able to assess these different populations. In some of our preliminary studies, we've already seen that polymer chemistry and nanoparticle characteristics such as size play a significant role in tissue tropism. Here I'm showing 2 of our PACE polymers, one incorporating a polyethylene glycol or PEG as a block copolymer. And using some formulation strategies, we've been able to generate nanoparticles of different sizes and assess their behavior in vivo. In this case, by IVUS imaging of whole organ fluorescence. And as you can see here, they have quite different biodistribution signatures just based on size with the same polymer chemistry. And looking at a wider range of polymer chemistries and sizes, we can again see these differences in physiological fate. One relationship that becomes apparent is that increased time in circulation results in increased opportunities to interact with extrahepatic tissues, as we can see here with PACE-PEG. It turns out that dosing is also important. And interestingly, we have found that increasing the dose of the same nanoparticle type increases the half-life. Moreover, we found that there seems to be a threshold dose beyond which you start to see significant accumulation in extrahepatic tissues, as illustrated in these plots of flow cytometry data of bulk organs and also specific cell types in the lung. And so our hypothesis here is that the filtration rate of macrophages is limited and that complete or almost complete occupation then allows for accumulation in other tissue and cell types. And similar concepts have actually already been discussed in the literature. And so, taking it one step further, we wanted to study the effects of intentionally either depleting or occupying macrophages on biodistribution. And our results so far are actually quite interesting and suggest that we are, in fact, able to modulate in vivo delivery by co-administering what we call biodistribution decoys. For example, here, if we take a formulation that normally only accumulates in the liver and spleen, and then combine it with pre-administration of one of our decoys, we can then achieve more widespread distribution to several different organs. Of course, in the context of CF, we're interested in lung delivery, and we were particularly excited to see that all of our decoys improve accumulation in the lungs. And so, to conclude, I hope I have demonstrated that we can deliver therapeutic nucleic acids systemically in vivo, and PNA-based gene editing agents in particular, to correct the underlying CF-causing mutations in multiple tissue types. I also hope I've shown you that in vivo delivery is a critical bottleneck for clinical translation, but also that we can take advantage of tunable polymer chemistries to design better delivery vehicles, and even modulate the physiological fate of nanoparticles to achieve higher accumulation in target tissues. And with that, I'd like to thank you all for your time and acknowledge all of the people who contributed to this work, as well as our funding sources. And I'd be very happy to take any additional questions. Thank you.
Very good, Dr. Piotrowski. That was beautiful. Thank you for presenting your data. The first question is, what is the amount of off-target editing with PNA?
Yeah, so we've looked at a number of potential off-target sites based on homology with a PNA binding site, and we've looked at off-target effects by next-generation sequencing. So we identify sites of partial homology, and in treated samples, we PCR amplify those regions and then submit them for NGS to look for any changes in base pairs. And so far, we haven't found any significant differences between our PNA nanoparticle treated formulations compared to blank nanoparticle treated formulations. We've found a little bit of variation, but it seems to be mostly attributed to sequencing error, because we also see that in untreated control samples. So far, we haven't seen any significant off-target genotoxicity.
Good, thank you. The next question is, how does IV administration end up correcting nasal and rectal tissues? Do you expect these LNPs to survive to cross multiple barriers? Do you know their half-life?
Yeah, that's an excellent question. So the half-life of PLGA is actually fairly short, but for the gene editing studies in particular, we do multiple doses. And in fact, the multiple doses allow for the PLGA almost acts as its own decoy, because in some cases the macrophages are occupied such that more tissues can be reached with subsequent doses. But we were actually sort of very curious of the same question. So I did some in-depth biodistribution studies that I didn't have time to go into in this talk, but we looked at microscopy of the nasal epithelium, the rectum, sort of all these tissues that we wouldn't necessarily expect to see a nanoparticle signal in, and we actually do, which we were excited about.
Okay. And in the interest of time, we're going to do just two more questions. The next question is, so your decoy strategy is very interesting. Have you tried that with other LNPs? Do you think that this could be a general strategy, or is it specific to PACE?
That is an excellent question. So we're actually not the first to think of this. There have been a scattering of papers, often looking at individual decoys and for various applications. I think we're among the first to kind of systematically look at that, but I do think it could be a generally applicable strategy. We're just trying to optimize it for our particular delivery platform.
Got it. Thanks. And the last question is, what is known about the cellular uptake of the NPs? Is there a receptor? Is it endosomal uptake and release and nuclear transport?
Yeah, so for the most part, with the main kind of nanoparticle that I showed here, polymeric nanoparticles. It's clathrin and caveolin mediated endocytosis. But we're actually also looking into sort of more specific mechanisms of what happens intracellularly and particularly endosomal escape, which we know will be very important for nucleic acid transfection efficiency.
Good. All right. Well, thank you very much, Dr. Piotrowski. That was great. So our next presenter is Dr. Catherine Excoffon, who's coming to us from Philadelphia. She's the Vice President of Research at Spirovant Sciences, and she's going to be talking about how SP101 delivery can restore CFTR function in airway epithelia. Thank you very much for joining us today, Dr. Excoffon.
I'd like to thank the organizers for the opportunity to be able to present our work on the delivery of SP101, which is able to restore CFTR function in human CF airway epithelial cultures and drive human CFTR delta R transgene expression in the airways of ferrets. This is our disclaimer. And my disclosure is that I'm a VP of Research and therefore an employee of Spirovant Sciences Incorporated. SP101 is a novel inhaled gene therapy designed to treat cystic fibrosis. We have several novel design features, including an AAV capsid that was selected for tropism to the apical surface of human airway epithelia. Inside of this capsid is the human CFTR delta R mini gene, along with regulatory elements, including a promoter, which drives mRNA expression, and a poly-A, which terminates the expression. Our planned route of administration is by single dose inhalation of SP101, followed by a small molecule called doxorubicin. We believe the mechanism of action is that our SP101 capsid has efficient apical entry into airway epithelia, so that's from the air surface of the airway. Once it reaches the endosome and is able to escape, doxorubicin has previously been shown in several publications to enhance the translocation of AAVs to the nucleus, where SP101 is then able to express the human CFTR delta R mRNA, which then translates into human CFTR delta R protein and is fully regulated and able to correct the CF defect in these epithelial cells. To prove that this works in vitro, we have shown this with several other donors, but in this experiment, we're showing well-differentiated human CF airway epithelia from a donor with class 1 mutations, meaning that they do not produce full-length CFTR protein. SP101 was added to the apical surface of these well-differentiated epithelial cells, along with doxorubicin. 1 week later, CFTR function was measured by Using chamber assay. On the y-axis is peak change in short circuit currents. As a control, we added the Vertex triple modulators VX770, 661, and 445. As expected for a donor that has no full-length CFTR protein, these modulators were unable to rescue any CFTR currents. By contrast, SP101 in the presence of doxorubicin at an MOI of 1,000, 10,000, or 100,000 was able to mediate currents which were dose responsive and similar in magnitude to those from epithelia from a non-CF donor. Of key importance, the SP101 without doxorubicin was unable to produce current, emphasizing the importance of doxorubicin in this approach. One question we've gotten many times is, what cell types does SP101 transduce? To do this experiment, this time we're looking at primary airway epithelia from a donor that had delta F 508 homozygous mutations. And we're using an SP101 reporter in that it has the identical SP101 capsid, but instead of carrying CFTR, it's carrying an M-cherry transgene. The SP101 was inoculated from the apical surface along with doxorubicin overnight, and a week later, 5-color immunostaining plus confocal microscopy was performed. I first want to draw your attention to this stain with the yellow cells. Each of these yellow cells represents a cell that was transduced with the SP101 reporter vector and is now expressing the M-cherry transgene. So greater than 30% of these epithelial cells were transduced. To look at cell type that were transduced, we co-stained the cells with alpha-tubulin for ciliated cells, MUC5AC for secretory cells, as well as HERX, which is in blue, showing the nuclei, and phalloidin, which stains actin. So in contrast to M-cherry transduced cells, our control cells had no staining in the M-cherry channel, but similar amounts of ciliation as well as secretory cells. I want to draw your attention to the XZ panel, where I've now labeled several cells with these yellow arrowheads or arrows that are stained with SP101 or with the SP101 reporter. Both ciliated and secretory cells are transduced, but the majority of the cells are non-ciliated cells, including basal-like cells. So this indicates that this vector has tropism to many different epithelial cells that would be important for correction for people with cystic fibrosis. The next question is, does it work in vivo? And in order to test it in vivo, we needed an in vivo model. The Engelhardt group has recently shown that SP101 capsid is tropic to ferret airway epithelial cells. Moreover, there's a CF ferret model which recapitulates the human CF lung pathology. Of importance is the ability to administer this vector by inhalation. So the approach was to give a single dose inhalation of SP101 through a plenum system with a nebulizer, followed by doxorubicin. Many different lung tissues were then isolated and investigated for in situ hybridization or human CFTR delta R mRNA expression. First, starting with the in situ hybridization, the data shows that SP101 vector genomes are abundant in many regions of the ferret lung. On the right-hand side, I have an example of the bronchial region as well as the alveolar region out in the lobe. We used RNA scope technology here with the probe directed to the sense strand, meaning it detects the viral genome. Each of these red dots is one or more viral genomes. And I hope what you can appreciate is that there's red dots in many different, both airway cells as well as alveolar cells, indicating significant transduction. To prove that this was specific for our vector genome and for the DNA, these epithelial cells were treated with a DNase to remove the DNA, and upon treatment, almost all, if not all, of the staining was removed, indicating the specificity of these probes. To look at the mRNA expression, we first asked the question, is doxorubicin important in vivo? To do this experiment, 10 different regions of the ferret airway were isolated. There was 1 sample from the nasal region, 3 from the tracheal region, 4 from the bronchial regions, and 2 from the lobe or alveolar regions. The data on the right is shown is an accumulation of all of those put into box and whisker plots, with the white line representing the median of the expression, the signal that's received from these tissues. And what we show here is that in the absence of any SP101 and doxorubicin, while there is some signal, it is below our limit of quantification, which is between 1 and 2. When we add just SP101 alone without doxorubicin, there is some signal. However, the signal is actually about 16 to 17-fold less than when we add doxorubicin, highlighting the importance and the significance of adding doxorubicin to this model system. We next wanted to know if expression is durable. And so we had samples from 2 weeks, as shown on the previous slide, as well as the end of our study, which was 12 weeks. And surprisingly and excitingly, while there was a trend to less expression, there was no significant decrease in the amount of mRNA expression in these animals. And I will note that in all of these studies, we used both reverse transcriptase positive samples, as shown here, as well as control samples that did not have reverse transcriptase. And there was no signal in the absence of reverse transcriptase, indicating that this is actually mRNA and not remnant viral genomes. Finally, and of great importance, we wanted to know, is there a difference between transduction in CF and non-CF ferrets? So these animals were given placebo or SP101 followed by doxorubicin, and 2 weeks later, the 10 segments were isolated for mRNA and expression analyzed. I will also point out that on the y-axis, this is a log scale. In the absence of SP101 or doxorubicin in both CF as well as non-CF animals, signal was below the lower limit of quantification. By contrast, robust signal was observed in both CF and non-CF animals, and no significant difference was observed, suggesting that there was the similar amount of vector entering into the airways of both CF and non-CF animals. So taken together, what I've shown you is that SP101 holds great promise for people living with cystic fibrosis. SP101 is able to functionally correct CF airway epithelia. Doxorubicin is required for this efficient correction of the cystic fibrosis current, the CFTR current. SP101 is tropic to many human as well as ferret epithelial cell types. Human CFTR delta R expression and CF correction are both dose responsive and durable. Finally, human CFTR mRNA expression is similar in CF and wild type ferrets, suggesting that the CF airway is not an additional barrier to SP101 as a therapy. Finally, I want to thank all the great people that we've been working with and are doing this work, including those at Lovelace Biomedical, the Cystic Fibrosis Foundation, Spirovation at University of North Carolina, University of Iowa, as well as Spirovant and our statistical consultant, Weizu. With that, I will stop and would be delighted to answer any questions that you have. Thank you.
All right, thank you very much, Dr. Excoffon. That was great, very exciting results. So we have a few questions. The first one is, what are the toxicity, side effects, implications, let's say, of using doxorubicin as part of the therapy here?
That's a really great question. And there was nothing of concern. These are preliminary studies. They weren't toxicology studies, and we have those toxicology studies ongoing in a GLP study right now.
Mm-hmm. Good. So the next question is also related to the doxorubicin. So do you see any side effects at the doses you use in ferrets? Do you know the mechanism by which it works in this context?
So the dose of doxorubicin that we gave these ferrets is well below what you would use in the clinic. And so the effects that you would see in humans, we don't expect to see the same kind of effects. And what has been published by the Engelhardt group is that it actually enhances proteasomal inhibition and escape to allow more of the vector to arrive at the nucleus and therefore get more gene expression from the vector.
Got it. Thank you. The next question is, so your ferret studies, some are included delivery to distal alveolar regions. Do you know the airways only contribution? So in other words, the airway versus distal delivery.
Yeah, that's a great question. That's exactly why we took different tissues from different regions in order to be able to understand some of that regionality. And it was using a plenum system that had the vector nebulized. So it was inhalation only, and we did not see as much expression in the upper airways as in the bronchial regions and in the alveolar regions. And so it was equally robust in the lower airways.
Good. And then one last question. CF lung disease tends to occur in a heterogeneous manner. Okay, so in diseased lungs, have you noticed any difference in efficacy, tropism, etc., depending on whether the region of the lung is injured or not?
Yeah, that's a great question. And so that's exactly why we did it in CF ferrets, because we wanted to make sure that there weren't any barriers in that CF lung that would further inhibit or slow down the transduction of those lungs. And what we saw was that the distribution was virtually identical. It was a CF model. It wasn't an extremely advanced CF model, but the fact that they were CF lungs, they had signs of CF, including no expression of the CFTR, and it looked good between the two. So we're very optimistic that this, especially since this vector was developed to be human specific, that this positive work in ferrets would be very positive for humans also.
Good. All right. Well, thank you so much for your presentation, Dr. Excoffon. Well, thank you very much. I'm really happy to see that the presentations today were really great, covered various different aspects of this really exciting area of therapeutics development. I'm really excited to see how everything progresses in the next few years. Again, I wanted to thank all of our presenters for taking their time to share their data with us. I apologize again for the initial technical difficulties, but thankfully everything was resolved and we finished on time. So thank you very much, everyone. And I hope you keep enjoying the conference and all the data that's being presented. Thank you.